Assalamu alaikum. This is Maria Yasin. From today, I am going to start O-Level Biology 5090 classes. And today, I am going to give you my first lecture of chapter number 1, Cell Structure and Organization. And my topic will be Animal Cell. Topics in this video to be covered are animal cell, its organelles, its components, and examples of animal cells. Okay, before moving forward, we have to know that what is a cell. So, a cell is a basic unit of life. Without cell, there is nothing which is present. There is no living thing should be present on this planet. So, there are two types of organisms. First is unicellular organisms. These are the simplest living organisms having a single cell. It means that this type of animals body consists of only one single cell. So all the functions they are going to perform should be performed by that only single cell. And the other type of organisms are multicellular organisms. Basically, these are the complex living organisms. They have many cells. So these many cells combine together to perform different functions, or many of the cells combine to perform a simple or a single function. So its example is human beings. So unicellular organisms, as you can see in this picture, paramecium, amoeba, bacteria and yeast are some of the examples of unicellular organisms. As you can see that the bodies of these organisms are single-celled. So each cell has its own organelles and they are going to perform different functions. And the other ones are multicellular organisms. As you can see in this picture, animals, human beings and insects, which are arthropods. So all of these are multicellular organisms. Their body is composed of many of the cells which combine to form tissues and then organs and then organ systems. So these are multicellular organisms. Now the components and organelles of animal cell. As these are the components and organelles of animal cell, components are basically are those which make the structure of a cell. For example, cell membrane and the cytoplasm are the components of animal cell, while the other cell vacuoles, nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria and centrioles and ribosomes are the organelles of animal cell. So, the first component of an animal cell is cell membrane. It is a thin layer covering the cytoplasm of the cell. As you can see in this picture, this purple color boundary which is covering a whole cell is known as a cell membrane. Its function is that it controls the passage of substances into and out of the cell. Most importantly, it controls the passage of water molecules. So it means that it controls the passage. It means that it is partially permeable. It means that it doesn't allow the large molecules to pass through it, or we can say that partially permeable membranes are those membranes which allows the movement of some of the molecules into and out of the cell while resisting or restricting the other molecules movement into and out of the cell. Like this, because of its partially permeable characteristics, cell membrane maintains the structure and its chemical reactions of the cytoplasm. That all the cytoplasmic biochemical reactions that are happening in the cytoplasm of the animal cells, so it is maintaining the structure of the cell and also the chemical reactions of the cytoplasm. It is the microscopic picture of plasma membrane. As you can see that it has double layered membrane and it has protein, pink color proteins are embedded in it, and and this is the whole structure of plasma membrane. Okay, the next component is cytoplasm. It is a jelly-like substance in which the metabolic reactions takes place. So this is a jelly-like substance which is enclosed by the cell membrane, in which many of the metabolic reactions, which are also known as biochemical reactions, that are taking place in it, and it contains nucleus and other organelles. So nucleus and other organelles are floating in this cytoplasm, and the cytoplasm along with the organelles of the cell is known as the protoplasm of the cell. The next organelle of an animal cell is cell vacuoles. These are the small membrane-bounded fluid-filled spaces. As you can see in this picture, that these two are the vacuoles. These are membrane-bounded entities, and these are fluid-filled spaces. Now, in animal cells, they store water and food substances, and they are usually not permanent. And in animal cells, these are more than one. As you can see that more than one vacuoles, are small vacuoles, are present in the cytoplasm of the animal cell, and these are usually not permanent. The next one is nucleus, the most important organelle. It is round in structure and has an other envelope around it, which is known as nuclear envelope. As you can see that this is just the structure of the nucleus. It has a nuclear envelope. At the bottom of the picture you can see the labeling of nuclear envelope. Then nucleoli or nucleolus, which is a darker color body present inside the nucleus. As you can see, and this dark color body in this picture, it is mentioned as nucleolus, and it is involved in the ribosomal RNA synthesis. This ribosomal RNA synthesis happened in this nucleolus, and when this is synthesized, it will move out into the cytoplasm, where it will make combine with the proteins and make a ribosome. Now, inside this nucleolus, inside the nuclear envelope, you can see that there are thread-like structures. These are made up of chemical DNA, which is deoxyribonucleic acid. So this is known as chromatin. This thread-like structure is known as chromatin. It is present inside the nucleus. Okay, now this chromatin material condenses into the chromosomes when the cell enters into the cell division stage, or when the cell is going to reproduce. This chromatin material will condense into the chromosomes. Okay, this is the a close view of cell vacuoles. As you can see, this is a nucleus. Nucleus is present in the center of the cell. In animal cell, nucleus is present in the center of the cell. Okay, then endoplasmic reticulum. Endoplasmic reticulum is a network of interconnected channels that extend from the cell membrane to the nuclear envelope. Okay, this network exists in two forms. One is the rough endoplasmic reticulum, and the other one is the smooth endoplasmic reticulum. Okay, the rough endoplasmic reticulum, as you can see in this picture, that on this picture there is a granular structure which is attached to it, which is making its appearance rough. Okay, what these granular structures are, let's see. That it has rough appearance due to the numerous ribosomes on it. So these granular structures are ribosomes which are attached to the endoplasmic reticulum. So it's giving it a rough appearance. So that's why it is known as the rough endoplasmic reticulum. Now its function is that it is involved in the protein synthesis. RER means that rough endoplasmic reticulum, and it is involved in the protein synthesis. Now the next is smooth endoplasmic reticulum. So after the rough endoplasmic reticulum, you can see the structure which is smooth endoplasmic reticulum. As it is shown by the appearance, there is no granular structures or the ribosomes attached to it. So that's why they are known as smooth endoplasmic reticulum. So it has a smooth appearance due to the lack of ribosomes on it. No ribosomes are attached on it. So its function is to synthesize fats and steroids. Steroids are chemicals, and transport of materials from one part of the cell to the another. So it is its function. Because it does not have ribosomes attached to it, so it is not involved in protein synthesis. So it is involved in the synthesis of fats and steroids and transport of materials from one part of the cell to the another part of the cell. So it helps in detoxification. It also helps in detoxification, like detoxification of harmful chemicals that are a cell ingest or taken by mistake. So it helps in detoxification as well. So Golgi apparatus. This Golgi apparatus, its structure is that a stack of flattened disc-shaped spaces surrounded by membrane. So it is a flattened disc-shaped spaces which are surrounded by a membrane. So it means that Golgi apparatus is also a membrane-bounded entity, and it is present inside the cytoplasm of the cell. So what's its function? Basically, it stores, sorts and modifies the substances made by the endoplasmic reticulum and package them in the vesicles to be secreted out of the cell. As you can see, from this side, it will receive the packages or the vesicles that are sent by the endoplasmic reticulum. These vesicles are sent by the endoplasmic reticulum. This Golgi apparatus will receive these vesicles, then it will sometimes stores it, it will sort it out and modifies the substances which are present inside these vesicles, and after doing this process, these vesicles will be secreted out from the cis phase of the Golgi apparatus. From the trans phase of the Golgi apparatus, it receives the vesicles from the endoplasmic reticulum, and from the cis phase, and these vesicles are secreted out of the cell and transported to the another part of the cell. Okay. So this is also the structure that all the proteins made in the rough endoplasmic reticulum depart in a membrane-bounded vesicles towards the Golgi apparatus, where the Golgi apparatus will receive it at the trans site of the Golgi apparatus and will sort it out, will modifies the protein, and then secrete it in the form of vesicles from the cis phase. Okay, now the mitochondria. Mitochondria are the small elongated organelles with folded inners. As you can see in this picture, these are elongated organelles and it has folded inners, and its function is that it carries out aerobic respiration. That it's a type of chemical reactions which happens in the presence of oxygen. Aerobic means that in the presence of oxygen. So it takes place in the mitochondria, and it is a process where the energy is extracted from the food in the presence of oxygen. And it helps in the energy that it extracted from the food, it gives to the other cells, other organelles which are present in the cell. So it helps in the cell growth and cell division. Now the next organelle is the centriole. It's a structure. So pair of barrel shaped structures at the opposite side of the cell. Basically it is present at the opposite side in a cell, and they play important role in cell division. Basically, as you can see, if this is a cell, these centriole structures are present at the opposite side, and during the cell division, its function is to produce or arise microtubule filaments from one side of the cell to the another side of the cell, like this. And why these microtubule filaments are arises from the centriole? It is to basically attach the chromosome structures in the middle of the cells, like this. So this is all about the cell division. It helps in the cell division of the animal cells. Now the next one and the last one is the ribosomes. As you can see, this is the structure of the ribosome. It has a large subunit and a small subunit which is attached to each other. and these are tiny granular structures floating in the cell cytoplasm or attached to the rough endoplasmic reticulum. So it has two subunits and are sites for protein synthesis. So these are the two subunits and these are the sites where the protein synthesis occurs. So how the protein synthesis occurs? Basically, the messenger RNA, it will capture the messenger RNA, attach with the messenger RNA and read the information which is present on the RNA and interpret that information and involved in the protein synthesis. Through this information, the protein will be formed. Okay. So some examples of animal cell. Okay, first of all, the liver cell. As you can see, the first picture is the you can say that okay, this is the first example of animal cell. It is a liver cell, and in the first picture you can see that it is a stained microscopic slide of liver cells, and this single liver cell has all the same structures as that of the animal cell, like it has a plasma membrane, which is the outermost cell surface membrane. Then it has a Golgi apparatus, free ribosomes, the most important nucleus, and then mitochondrion. Mitochondrion is this word is used for a single mitochondria. Okay, so when we are talking about many mitochondrion cells, so it will be mitochondria, and if we are talking about a single one, so it will be called as mitochondrion. So the cytoplasm and the rough endoplasmic reticulum and the smooth endoplasmic reticulum as well. Okay. Now the next one is cheek cell, and the first one is the stained picture or the stained slide of cheek cells. It has the nuclear membrane, it has a cell membrane, cytoplasm and the nucleus, as you can see in this picture. The next one is red blood cells. The first diagram is from the microscope. It's a stained one, and the next one, it has no nucleus. The red blood cells doesn't have nucleus, and this is the diagram of the red blood cell. The next one are the muscle cells. There are three types of muscle cells: the skeletal muscles, the smooth muscles, and the cardiac muscles. Skeletal muscles are basically they make our muscle cells like bicep and tricep muscles, and it has shape like this. And these smooth muscle cells have different shape from the other two muscle cells, and these smooth muscle cells are basically making our bladder or basically intestine, or most importantly our stomach muscles are made up of smooth muscle cells. Okay, then the cardiac muscles, these are different from the other two. So the basic reason is to show these type of cells. Basically they have different structures, so you have to know that how these cells looks like. So thank you for watching my video. Hope you liked it, and in the next video I will explain the plant cells.